so this is topic 12. It's all about mitosis and cell regulation, cell cycle regulation, and apoptosis is what we're going to be talking about. So where do we start? Okay, let's start from interface. We talked about interface in the previous videos, but the first is the interface, and the second one is the mitotic phase in general. So there's two steps, two big global step. Global step 1, interface. The second global step is called mitotic phase, which is shown right here. And there's the G1, the S phase, and the G2 phase in the interface, and that's the 95% of the cell cycle, and 5% of the cell cycle is the So like we said, G1 is all about growth, organ synthesis, replication, synthesis of regulatory proteins, increase in size, and the synthesis phase is all about chromosomes that are copied to two sister chromatids, and both have the exact genetic info. There's an exact copy, and these are connected by centromeres. They are homologous chromosomes. And the G2 phase is all about the final growth of the interface. So this is the final stage of interface preparation for mitosis. So centrosomes are replicated. Very important that centrosomes are replicated in this stage. It's the G2 that's separate, that they duplicate centrosomes, and the fiber, well, the spindle microtubule fibers are formed to separate the centrosomes later on in the mitotic phase, and to perform the mitosis and other cell divisions as well. Yeah, so recall that the somatic cell and gamete cell, somatic cells are the body cell, gamete cells are the eggs or the sperm, and these are produced in ovaries or testes. These are produced from these ovaries or testes, going through meiosis. And they are haploid. They are haploid for the one set of chromosome from one parent, and diploid for two sets of same chromosome, one from mom, one from dad. And globally, there's a mitotic phase. In the mitotic phase, there's a meiosis and the cytokinesis. Cytokinesis is about the dividing, so formation of cell wall in the plant cell, or formation of I forgot how to call it, but microfilaments are going to cut the cell into two after the division. And mitosis is the process of the chromosome replicating, dividing, etc. So here is the process of mitosis, and these are eukaryotic mitosis. Don't really care about what type of mitosis, I guess. Nucleus will double, and in general, nucleus will be doubled. So that's the whole point, to copy the genetic information and multiply chromosomes, and copied on both sides of each chromosomes before they cut, which is a cytokinesis. So interface is what we already talked about earlier, about the G1 and the synthesis, and the G2 phase, where they will replicate stuff, they will produce proteins, regulatory proteins, divide chromosomes into two, and they get bigger, and they will prepare for mitotic phase because the centrosome is already doubled in G2 phase. So in the initial state, right before mitotic starts, there's already two centrosomes. So there are 5 phases in the mitosis one: prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinesis. So the 5 major ones. The first one is the prophase, which is chromatine turning into chromosome, and nucleus will disintegrate, so nucleus will disappear, and nucleolus will disintegrate, and nucleus is still intact. So the inner nucleolus will be gone, but nucleus itself is still there in the prophase. So chromosome is duplicated, I guess I could say in G2 or prophase, I guess, and microtubule spindles will be formed. That's kind of contradictory. We said that was in the G2 phase, and they will stretch apart. They will stretch apart the cell. These spindles will stretch apart the centrosomes, which will also stretch apart the cell. So we said that these ones are called the asters. I think it was asters. It's a spindle of microtubules that originate from centrosomes. It's just a terminology we should probably know. And the prometaphase is a nuclear membrane that breaks. So nuclear membrane, which was not broken in the prophase, will now break. So chromatin turned into chromosomes in prophase, which is important. And then nuclear membrane will now break, which was not the case in prophase. So after they are broken, centrosomes will go to the opposite side now, so they are going to spread completely like this. And the microtubules will attach to the centromeres and form kinetochores. So kinetochores will be formed at this spot here, which is the centromere of the sister chromatids, and these are this is called the centromere region, and these are these are proteins assembled at the chromosome centromere to link the chromosomes to the microtubule from the mitotic spindle. So it's really, these are proteins, these red ones are proteins that will link the mitotic spindle to the chromosomes, to the individual chromosomes. And this will happen during both mitosis and meiosis. And this is what it looks like. This is a chromosome, and they're motor proteins inside the kinetochore. There's the microtubule, and that's going to be separated. So microtubules are good because they're dynamic and they're able to stretch and to go grab and to shrink later on to separate them. So it's a dynamic instability. I think it was dynamic instability. And there's no more nuclear envelope. So 3 is the metaphase, and metaphase is the lining up. So they will line up metaphase plate, cell equator, metaphase plate of the chromosome. So these are all homologous chromosomes that are just lined up. They're just chromatids, sister chromatids that are lined up. Yeah, so they will form these X-looking chromosomes, and they will all line up like this. And each side is, well, the exact copy. These are sister chromatids, and sister chromatids are going to be broken into half, into each side, to the opposite poles. And yeah, so centromere will break, sister chromatids will be separated, and there you go, we have the same identical genetic information DNA on each side. And they are still diploid. They are going to be cut in half, but they're still diploid because this could be the mom, this could be the dad, this could be the mom, this could be the dad. So like they're not separating the parent pairs. They're only replicating, and so like duplicate themselves and cut it in half. So there is no separation in the diploid to haploid process here. Yeah, so very important. When they separate, they are called daughter chromosomes. Daughter chromosomes after they separate at the anaphase of mitosis. Telophase is the ending of the mitosis. So the chromosome will become more loose, and they will turn into chromatine fibers. Will loosen and become less coiled, and the spindle fiber will be gone. So the spindle fiber that were pulling them will disappear. The nuclear membrane will reappear, and the formation of so nuclear membrane will form around these nucleuses. So we can see that the nucleus is still there in prophase, and they will be gone from prometaphase. From prometaphase, metaphase, anaphase, all the way to telophase, there is no nucleus, and nucleus is starting to be formed at telophase. And this is the end of mitosis. But the division is not entirely complete yet, because cytokinesis has to happen. Cytokinesis is the final cutting of the two daughter cells. That will leave a cleave furrow, and these have identical genetic information in them. Yeah, it's a contractile ring of microfilaments. That are going to cut them. And I think there was a myosyne as well. The myosyne and actines. Yeah, actines and myosynes, proteins. Either way, there's a microfilament that is going to cut it. So overall, interphase is from single to doubling itself, and mitosis is about separating these doubled chromatins into two. That's the general process. Now, finished talking about mitosis, we're gonna go to the GO phase. GO phase is between the M phase, mitotic phase, and the S phase, synthesis phase. So it's all about inactive state. cell will go into a non-actively dividing state, such as liver cells and brain cells, so neuron cells, that will still do their function. It's a common misconception that telophase is that the cells are sleeping. They're not sleeping. They're totally active and they're still doing their jobs, functions in the body, but they will just not be dividing. The whole point is that they will not divide. And if they do come back dividing, they will go through the G1 phase again and repeat the cycle. So now cell cycle control is what we talked about in the G1, that there is proteins that are going to be synthesized, regulatory proteins of each cycles. So the cycles has to be triggered by a signal, or different signals from hormones. I don't think it's hormone. It's a regulatory protein. So regulatory proteins will give signals for each stage to be giving checkpoints. So they go like, okay, yes, G1 can be started or G2 can be started, etc. And these signals are important because if it does not come, well, then that means they will not proceed. So the most important is the G1 checkpoint. It's called a restriction point. And it's a restriction point most important because after G1, S phase, G2 phase, and the M phase will proceed. So the G1 is one of the most important checkpoints. So they are called cyclical operations, which is the checkpoint of cell cycles. Yeah. So if they do not receive one G1 protein, they will go to a GO phase, which is the phase that they do not divide. So cyclins and cyclin-dependent kinesis are the two regulatory proteins that will promote the path through of the cell cycle. So cyclins and cyclin-dependent kinesis. And there are many types of cyclins, and each cyclin will be higher in concentration for each dependent time. So like at time S, a synthesis phase, there's the E that is higher. At time G2, A is higher, etc. And so CDKs are always kind of sleep. They are inactive and they have to be activated by the cyclins. And when they are activated by the cyclin, they will bind to a target protein that is inactive, and that is going to be activated. And yeah, so the cyclins are produced and they will be gone because the concentration has to vary. So the ones that are produced is the cyclin, and cyclin-dependent kinesis are just proteins that are already existing, I think, in the body. So if this was not properly regulated, there's going to be tumors, because if they are not regulating the regulatory protein, they do too much cell division, too much cell division, which leads to tumor. So P15 is a transcription factor protein, and what could suppress tumors. So they will break a cell cycle. They'll stop a cell cycle from happening if there's a mistake, or if the DNA is required of a repair, they need to do a pause, etc. They will suppress the cell from dividing, and this is important for suppressing tumors, which is wrong cells from overgrowing randomly and crazily. Some are predisposed of cancer because they're born with mutated P56. So if P56 is mutated, they're prone to cancer. And because the second mutation of P53 is directly, you're doomed to cancer if there is a mutation of P53 for a second time. So that's why some people are vulnerable to cancers when they're born. So apoptosis. Yeah, so P53 is what suppress tumors, but also if there is not enough, if the repair, like I said, that needed DNA could not be performed, so if repair is not possible, the cell will die, will kill itself, called apoptosis. Apoptosis is a programmed cell death that is done because there is no way to treat it, and if it cannot be treated, the body will not copy damaged cells, so they will go through apoptosis. But if there is not enough apoptosis, if it is not performed, what happens is cancer, because dividing will continue forever, and these bad cells that are supposed to die will not die and keep dividing, leading to cancer. So apoptosis happens to over 50 billion cells in the body, which is a number I think we should be knowing. Yeah, so more than 50 billion cells of the body, they're dying. So they could be triggered. These deaths called apoptosis could be caused by triggers such as heat, radioactive radiation, low oxygen, so if you don't breathe, signals from the virus. So these are stuff that was covered during the virus, how they can kind of hijack host cells and tell them to do things that they're not supposed to do and kill themselves, and signals from other cells as well. So that's where I think cancer comes from, or cancer or other diseases telling the neighboring cells and the cell in all of your body to kill itself, and they will do that. And one of the consequences of too much apoptosis is Alzheimer. So Alzheimer comes from too much of cell apoptosis. Too much cell dying is Alzheimer, but in the contrary, too less of cell dividing is cancer. So apoptosis also occur in the embryonic development. So it's not something that will only happen after the child is born. It is something that could happen even during embryo stage of life. Embryo stage of life, apoptosis as well. So what happens if they were triggered? So the process of apoptosis is that they will be triggered by external signals, and they will activate proteolytic caspases. I think when you can do that, they'll break peptide bonds. So that's essentially what's happening in peptide bonds, which is the protein bonds. So all the proteins in the cells are going to break, and what's happening is that they will bleb and become really smudgy and melting kind of looking, and the fragments of DNA will be broken, and the cell itself will just bleb and become a smaller fragment, and everything's going to be a smaller fragment, and therefore they will kind of explode like this. Not explode, but melt, and send signals to the white blood cells so that they can macrophage or phagocytosis of these garbage, the déchets that happened. Yeah, so macrophage is a type of white blood cell, and phagocytosis is the process of eating that's gonna happen. So macrophage are the white blood cells. Right? Do you know?
I wasn't listening.
Macrophage is a white blood cell. Oh yeah, you did it last year, forgot, whatever. So apoptosis is the normal healthy way of dying because the cell is programmed to die, whereas necrosis is a disorganized, unplanned bursting of the cells. So it's a sudden inflammation that could cause harm to the body, other parts of the content, and that is spilled into the ECM. So misregulating other cells will harm other cells. That's necrosis. It is a disorganized, unplanned bursting. So, like I said, with apoptosis, too much will be Alzheimer and death of the cells, and too little is cancer. So what is cancer? Is a unchecked profile of damaged mutation of cells that are that go through continuous cell cycle and ignore cells signals to stop them. They will not go through apoptosis. They can also give signals to other cells to do things that they're not supposed to. These are the cancer cells, and they will rapidly divide and cause tumors, spread all around the body, and that's why cancer could be deadly and very, very scary. And these days, cancers are prevalent, so there's a lot of techniques and science and technology that will prevent cancers, and they are prone to older people still to this day.